Our talk tonight is by Dr. Angel Medina. He is the reader in food mycology at the University of Cranfield. He has many other titles and awards, but right now, all he cares about is mycology. His talk tonight is making fungi travel through time to predict future food safety problems. It's very exciting. I'm really excited. I hope you're excited too. And without further ado, I'm going to hand over to Dr. Angel Medina.
Thank you very much, Nathan. Can you hear me properly? That's good. So the first thing I would like to start with is thanking the BMS really to give me this opportunity that I always call like a window to the world. So it's a fantastic opportunity for me and for the research group that I represent here, the Applied Mycology Group, to kind of show some of the work that we've been doing in the last decade or so. And then I would like really to thank everybody that has made it and you that are at home spending few minutes with us to learn what we have been doing in this time. So yes, the talk today is going to be making fungi travel through time to predict future food safety problems. So the outline of the talk, I'm going to try to introduce a bit what are we talking about, what do I mean when I say time. I'll speak a little bit about climate change and the factors that will affect fungal pathogens and what will be the implications for food safety. Then we will start going up and down in time, and I'll ask you to concentrate and you know, like have imagination in your mind. And I will show you the different results that we have been getting. And then finally, there'll be a couple of slides where we will be able to discuss about, you know, the main problems, and I'll try to give you some food for thought for the coming days. So let's start with no more further ado. And this is the timeline that we are going to be using today. I'm going to focus mainly in 4 time points, and let's start by now. You know, now is today is 3 months ago, and what we can see now and what we see around us now is that we recently had here in the UK COP26, and what we see is that more and more we are more aware of climate change and the problems that climate change is producing around the world. There were news about, you know, how warm is it, the weather is getting in the COP26. There were alarms. You know, although everything we appear to be doing about climate change, we still, you know, in the range of the 4+4 degrees scenario by the year 2100. Then we can see more and more reports everywhere in the news about, you know, although the pandemic, the global CO2 emissions are shooting back to record levels, and you know, the people was expecting for it to be lower because of the decrease in economic activity, but no. The Guardian has raised the climate crisis here in the UK with some phenomenon that we haven't seen. You know, last December has been one of the warmest in the last 65 years. So that tells you everything. And then we have other places that instead of being too hot or too cold, or they have floods. And colleagues from the Imperial College have, you know, published recently in last year a nice report on how, you know, there is going to be floods, heat waves, droughts, wildfires, crop failures, and these type of events are becoming more and more common year by year. But I think, you know, let's have the first big question about what is happening and what are the main environmental changes that are going to affect, you know, the main subject of this talk, which is going to be fungi. So those are going to be in one side, in this graph, you can see the predictions for the increase on emissions of CO2 in different scenarios that were given by the International Panel for Climate Change very recently in 2021. And we can see how in most of the scenarios, mainly the two reds, is going up, and there have been some projections about that. The other big factor that we have to consider is, as we said, that temperature, and most of the predictions, and in COP26 there was a big debate about, we are going to the +4 scenario if we don't change things. So quite hot in the press at the moment. Another thing that is going to happen, and it is that it's been predicted that because of the changes in temperature and other factors, the temperatures of the waters in the oceans, the way it's raining is changing and will change in the future. So, for example, if you look at this map created by NOAA, there will be most areas in Europe where there will be less rain, including the south of the UK. And the problem is that this is average, but it doesn't take into account that it might rain everything in one day, or it might not rain for a few months and then rain during a very shorter season. So these averages are even worse when you look at local, where there might be big changes in small areas. And then another thing that is going to be important, and it is already being important, is that colleagues from Exeter, Dan Weber and colleagues, have published a series of papers in the last years where they have described how plant pathogens and pests are moving from the equator to the poles at a rate of around 5 kilometers per year. So that is going to make, some fungal pathogens appear where they were normally not present, or is going to give the opportunity for growth and the environmental conditions to grow in places where they normally were, but they were not able to produce the disease. So a lot of things are changing. So making a little bit of a recap about all these things that we've said about climate change, and to make it easier for the presentation, we are going to say that there are going to be three main factors that we are going to consider in the talk today. One is going to be the rain, how much rain we have, and I also put the sun there because of drought. Then it's going to be an increase in temperature, and there is going to be an increase in the CO2 that we have in the atmosphere. So, okay, so now let's focus on these three parameters. So let's talk again about our timeline, and let's go back to now. And how do those look like now? So this is the data for the UK, published for 2021. And the provisional data about rain says that it only rained 93% compared with the average in the last 30 years. Meaning that year by year, what we are seeing in general when we look at this data is that it rains less than used to rain. When we look at the temperature, in this year we've had 0.1 degrees Celsius above the average as well. So, and with a mean of 9.3. And when we look at the global average of atmospheric carbon dioxide in 2020, it was 412.5 parts per million. And that was, in 2020, one of the biggest increases in the last 75 years. So it appears that year by year, we are going to a scenario where we have drier, we have hotter, and we have increased CO2. So how does that future look like? So when we put in numbers, those predictions for the ICCP and what they are saying, how will these values look like in the year 2100? So what is going to happen is that it's going to have less rain, and we'll have more drought episodes. We're going to have increases in temperature that the current models show increases between 2 degrees and 5 degrees. And the amount of CO2 in the atmosphere is expected to be around year 275 and 2100 is expected to be between 650 and 1000 or even higher. So now the question is, if we look at those conditions in the year 2100, will these conditions have implications for food safety? So again, this talk today is going to be about the research that we've been doing with regard to mycotoxins. In the Applied Mycology Group, we do a lot of ecophysiology on this toxin, on this fungi that produces mycotoxins. And what we do to these fungi is we mix different environmental factors to understand how these environmental factors affect the way they grow and the way they produce these toxins that will end up in our food. And then we do a quite unique approach that we call molecular ecology that I'll come back later on to explain a little bit better. But now for those that might be now a little bit opening their eyes and thinking, oh my God, what are mycotoxins? I put here a small explanation about them. So we are very interested in mycotoxins because mycotoxins are naturally occurring toxin metabolites that, you know, the molds, while they are growing in the field or they are growing at home, they can produce this type of metabolites that will contaminate our food. And then when we eat them, they might be able to produce us disease. These are very interesting because they are very heat stable. So they are very difficult to destroy. With other type of compounds, what will happen is that if you cook, if you boil, if you roast, they will disappear. However, mycotoxins, if you have them there, it's very difficult to remove them from the food item. And then one other characteristic that they have is that they are produced by very, very, very common fungal species and genera. And I'm sure, you know, if we will take a swab and analyze the fungi that we have in our shoes at the moment, almost guaranteed that we'll have some of these species in our shoes. Some other considerations about mycotoxins is that they can occur everywhere. As I said, from the field to us at home by not storing things properly. While we are storing, while we are processing our food, anything could happen. And then another misconception that a lot of people think is, you know, that they can detect when there are mycotoxins in their food, and they are very difficult to detect because, first of all, that there is no visible mold doesn't mean that the food is safe. And also another thing is that they are not visible, you can't smell them either, and they have no taste for our palate. So really, if you have them in your food, you don't know that they are there. So you have to be very careful, and you just have to trust scientists and the authorities to have control them in your food. One thing that makes these toxins very interesting for us is the way they are regulated within the fungi. And what happens is that, as I show here in this graph, most of the genes that the fungi needs to produce these toxins are all together in the genome. And normally before these genes, you have a regulation area in the genome. And it has been described in the literature for many years that many of these environmental factors that we have been talking about as the main ones about climate change, water activity, pH, temperature, CO2, light, we know that they have an effect on the way that these groups of genes that then lead to the production of the toxin are regulated. And now I'm coming back to the term of molecular ecology, because what we like to see is whether what we see when we do those modifications in the fungi, we can relate it back in the way that these genes have been regulated in the fungus. So we modify the conditions, we see what happens in the fungus, and then we go deeper and see whether the genes change to produce that effect and to understand why we could see those changes. So now let's focus it a bit more. Now that we all are in the same page and you know what mainly I'm going to be talking about, now let's see what food are we going to be talking about. And we are going to be talking today about maize and a fungus that is called Aspergillus flavus. Why maize and Aspergillus flavus? Very easy. In conditions that we will describe as now, and you know what it has been the norm for many, many years, the species that will normally dominate the mycoflora in maize plants is Fusarium verticilloides. So you will normally find, you know, your maize contaminated with Fusarium verticilloides. Fusarium verticilloides produces a toxin, a mycotoxin, but you know the amount of toxin that you need to eat to really get the disease is quite high. However, if we have drought episodes or we have increased temperature, then the problem is that Fusarium verticilloides cannot grow effectively anymore. And what happens is that Fusarium verticilloides gets substituted or gets removed by Aspergillus flavus, that under those conditions will be able to grow much more efficiently. And that has a problem within it. And the problem is that Aspergillus flavus produces the mycotoxin called Aflatoxin B1. And Aflatoxin B1 is the most potent carcinogenic molecule of biological origin known so far. I will repeat it because this is very, very important. It's the most potent carcinogenic molecule. It produces hepatic cancer, and if you eat enough of it, you die because of fulminant cirrhosis. And it's been described as carcinogen group 1 by the International Agency for the Research of Cancer. This has led that there is very, very, very strict legislation on the maximum levels in different foods in the EU and the UK. And the main places where you can find this is in maize, in cereals, in dried fruits, and in your nuts. So if you are now at home with a nice glass of beer, having some pistachios or peanuts, don't worry, keep going. We hope they don't have this. Then the effect, so then what has happened is that in the last 10 years, there has been a lot of interest and there has been a lot of publications on how different environmental conditions can modify the production of aflatoxins in maize. And most of our colleagues around the world have looked at combinations of two main parameters when looking at this problem. And they looked at combinations of rainfall that can be associated with what we use when we are looking at food items, which is water activity. So the water activity is related with the rainfall because it's the available water that you will be able to find in a food for the microorganisms to grow. Okay, so when I have rain and my food item gets wetter, then there will be more water available for the microorganisms to grow. And then I might start having a problem. And then another one that they have used is temperature. So now the question is there any data already available? And yes, there is many data available, but I'm going to highlight a specific work from our colleagues in Italy, Professor Paola Battilani and colleagues. I think we have Marco Camardo as well in the talk. So authors of this paper, and they used temperature and meteorologic data in general, temperature and rain, to project how much or to project the risk of having aflatoxins in Europe. And what you can see here is that in scenarios going to year 2079, they model the +2 degrees and +5 degrees scenario. And what you can see here is that with +2, we can see the increase in red spots in the map. So compared with the baseline that will be now, with +2 degrees, we'll have more red. When it's redder, that means that the increase is an increased risk to have aflatoxins. So with the +2 scenario, it remains quite restricted to the south of Europe. However, look at what happens when they model the +5 scenario. There was a much higher risk of having aflatoxins. However, it looks like the red is much more spread, but what is very worrying is that you can see that much more area has color, meaning that many more countries and in many more places there will be the possibility and there will be the conditions for these fungi to produce this very nasty molecule that will contaminate our food. And even, you know, we can see here a little spot in the UK, which was quite surprising for me when I first looked at the paper. So now let's go back to our timeline and what has been our different approach. So I'll say that, you know, now we go to year 2009, and I'm not going to mention names, but in 2009 there was this enthusiastic postdoc coming from Spain that you know, went to the UK with the idea of learning from a professor in Cranfield University about how different environmental factors were affecting the growth of fungi. And having multiple chats between them, they decided that it was kind of timely, because of all the things about climate change, to, instead of looking only at water activity and temperature, to include CO2 in the picture, because it was more and more evident that that was going to be a big problem. Then what we were doing and what we were planning was really to replicate the conditions that we'll have in 100-year times approximately. So then it was a matter of forward to the future. So we decided to send fungi and put them under the conditions that we'll have in 100 years time and see how they were behaving. How did we do that? We did exactly what was suggested in the predictions. We took food items, in this case maize, and we use an intermediate water activity. We use the different scenarios, created +2 and +5, but we added to the research that was being done at that moment the increase in CO2 conditions. And we did this in environmental chambers that were sealed, so we could control exactly what was the environment inside those boxes that we were using. And these were the results. And here what you have is different temperatures at the beginning, 30, 30, 30, or 37, 37, 37. We have 7 degrees of difference. Then 0.91 is dry conditions, 0.99 is wet conditions, and then we have 350 ppm of CO2, 650, and 1000. And what you can see is that in all cases, in all conditions, mostly the general trend was that when we included CO2 in the picture, the amount of toxin produced by this fungus was increasing. In some occasions, the increase was 99 times higher. So it was a big concern, and this helped us to, go to different conferences, and, then we got a little bit of money to study the genes, and we could relate those amounts of toxin and those increases of toxin in certain conditions with with one of those genes that are regulating the set of genes that was one of the regulatory genes. And again, we got invited to a lot of conferences with that. And then we come to this moment of serendipity, where one of those moments that I would describe as career changing. And, it was back in 2014, and, we went to the World Mycotoxin Forum, and we were awarded the prize to the best poster. After having a poster on climate change and all the data that I've just shown you, and the prize for that poster was a bottle of wine. So there you find, two people from the UK in a different country with hand luggage, flying with Ryanair, and not being able to bring the bottle of wine back. That was the price, no money, by the way. And then the question was, so what do we do with this bottle of wine? And then we decided, what any person, with a bit of pragmatism would have done. We went to a bar just in front, asked for some disposable glasses, opened the bottle, and have the bottle with the people that was remaining there in the room, the people that didn't left running, with the good luck that, we had some people, they're very interested in Aspergillus flavus, and we started chatting. So what have you done? Very interesting, your results. And, I've realized when we were sharing those glasses of wine that it was people from the USDA and from North Carolina State University, which were the two groups that had published for the first time the genome of Aspergillus flavus. In the pack also, we had people from Boku in Austria that were really, the experts on multi-mycotoxin analysis. And then, chatting about, do you have the samples? Yes, oh yeah, let's collaborate. And there comes that question, from the USDA and North Carolina, have you done RNA-seq in your samples? So we said no, because we don't have the money. How much does it cost? They say at that moment, it was like around, for the samples that we had, it was around $80,000. I said, well, we don't have the money. And I don't know whether it was the wine or the good environment, but they say, oh, but I have the money to do it. Send me the samples and I'll do it. And the same happened with, the metabolites. So that made, like over a glass of wine in a very innocent conversation, we had that moment in which we launched really the potential of our results and our data by including RNA-seq analysis and metabolomics. And, we had to go back to the future. We had to, we redid some of the analysis. We did the extractions for the RNA. We repeated the experiment like 3 more times, created the samples. And that gave us the opportunity of, instead of analyzing these 3 genes that are the ones that we had looked at before, we looked at the whole pathway, everything, to understand whether the CO2 was making an effect on a particular gene. Was it something general that was happening that was leading to that increase in the amount of toxin produced? And this is some of the data that we had, but put in a very simple heat map. And I'm going to draw your attention in these 2 green boxes. This first green box here, which says 350, is the conditions now, or in reality, the conditions that we had back in 2014. And this one, very different color, is exactly the same conditions, but increasing the CO2. So we could see that the effect was a very general effect, not affecting only single genes, but it was something bigger that was affecting more than just a couple of genes. And with that, we were also able to collaborate with bioinformaticians in the USDA, and they helped us to create a new system to study that data that we were creating, but not only for aflatoxins, but also for all other secondary metabolites that were produced by this species. And they created a new mathematic approach to study the three-way interaction, because normally with RNA-seq, the comparisons are one against another, but it was difficult to have a multiple comparison. And we saw, when you look at this slide, we saw that when you look at one of the clusters, that is the cluster for aflatoxin, in the last column, that is the three-way interaction, we saw a very positive interaction, 2.9 here, which was higher than 0, meaning that really when we have CO2 in the picture, the way in which water and temperature interact between them changes completely. And we found very interesting that we found even higher figures for other compounds. And one of them is also regarded as a mycotoxin. And it's also something that we would not like to have in our food. And what happened is that then we used the relationship with Boku in Austria to check whether is there any difference in this metabolite when we use the CO2. And yes, indeed it is. When you look at conditions with high temperature and 400 ppm of CO2, which is this line here, 37,400, this is the amount of cyclopiazonic acid, this molecule that we are describing now. And when we modified it and we included the CO2 to the picture, you can see how the proportion and the size of the yellow part of the line is. So we could see that with our approach on looking at the genes and using this new data, we can predict that there is going to be problems and some other components or some other toxins might become important when we are moving further on time. But obviously, you know, we published few papers about this. We've had very nice conversations with a lot of people that is already in the call today, but we hit what normally researchers will hit very commonly. And it's what I like to call the reviewer number 3 conundrum. What do I mean by this? Is like you have submitted this paper to a high impact journal. First, a reviewer will say, brilliant, you know, this English here is not good, change this, blah. Second one, very nice, very timely. Third one, complete disaster. This is just not publishable. And the main criticism, or one that really, you know, opened our eyes and help us to move the research forward, was some comments about, you know, that it was good to have a prediction, but they were not trusting that could really represent reality, because we were moving fungi from now to 100 times later in time, in the future, directly. And then, you know, we looked at possibilities and looked at what other colleagues were doing, and they were acclimatizing the strains. So what does acclimatizing the strains, and what will happen? What will happen if we acclimatize those strains? So let's go back to our timeline. So what we were doing before is going directly from, you know, from this area of the graph to the other side of the graph in one step. And what the reviewers suggested was instead doing it in very little steps. That will mean that instead of going directly from current to conditions in the future, we'll be subculturing the fungi in increasing temperatures and increasing CO2, in a way that we will arrive to the final conditions. And really, it was a nice suggestion, so we didn't doubt that we had to do it. And I'm going to show you some of the newest results that we have in that regard. So here we have some Aspergillus flavus that in this case, we isolated from pistachios. Same problem in pistachios than in maize. We can have the contamination with Aspergillus flavus. And then we looked at the growth of acclimatized in blue, with non-acclimatized in blue versus the acclimatized for 5 generations until we arrived to 37 degrees and 1000 ppm. And what we could see really is in two different strains coming from the pistachios, that there was not really a significant change in the way they grow. They still look very similar in the plate. You look at the fungi, they really don't show a lot of difference between the ones that were in the now and the ones that are in the conditions of a year in 100 years time. However, the picture changes a lot when we looked at the aflatoxin production. They look the same, their growth was exactly the same or very similar. However, if you look here, the control is the blue, the non-acclimatized is the orange, and the acclimatized by 5 generations is the gray. And surely you can feel quickly that there is a lot of gray in this graph. And what we saw is that in most cases, acclimatized strains were producing higher levels of the toxin. That is no good news, because if we look back at the research that we did, we have already described that it produces more when you have the CO2. It will be even higher, or data points out that could be even higher if we have the strains, acclimatized during time. And all that map that we showed from our colleagues in Italy was based on only water activity in rain and temperature. So what will happen if we add CO2? We'll have much more red, and for sure the red will be much more northern. Maybe it could arrive even to the UK. And now your following question is, you might think, oh, this guy is obsessed with Aspergillus flavus, and it might be because he found, the way of making his career, and develop all this work about Aspergillus flavus. But this might happen only with Aspergillus flavus. So the question is this only happening in Aspergillus flavus? And the answer for you is no. And we've done this similar type of work with different fungi. In this case, for example, with another fungi that produces another toxin, which is called Microtoxin A. And although some of the changes are subtle, but when we increase temperature and we put them under stress, you can see that under current conditions, they won't produce almost any toxin. But then when you put them at 1000 ppm, meaning 100 years from now, they start having some toxin produced. And in general, you can see that although there might be not a lot of difference, you have points where there is a very marked increase. The same happens with Aspergillus carbonarius, which is also in grapes and responsible for the contamination of wine with Microtoxin A. When we look at the orange here, in some cases, depending on the conditions, we can also see that orange might be higher depending on the conditions that we are trying to impose. And finally, very important for those eating porridge every morning for breakfast, be careful with your oats, because oats are contaminated with Fusarium lancetiae that produces also a couple of mycotoxins. And what we've seen is that when we put them under climate change conditions here in the UK on, British oats, we have increases of, 5 times to, around 5 times, and, in some cases, even 200 times, because it goes from very, very little to a considerable amount. So not something that only happens in Aspergillus flavus, and much more room for research and to keep working in this area. So now we are coming now to the end, and I think the big question is, what have we learned in all these trips going back and forward to the future with our fungi producing mycotoxins? And these are, I think, those take-home messages and some food for thought. So there is going to be, the question is going to be an increase in plant diseases, and the production and pest production and the interaction with plants might increase the mycotoxin contamination? And the answer we have already demonstrated that is yes. There is a significant potential impact on nutritional value of the food because of not only pests and more diseases, but also because they might have concomitant contamination with mycotoxins. And the answer to that appears to be yes, when we go to the future. We know that there are metabolomic changes. So as I explained, there are changes in the species that produce when they should not be producing, or when they are not producing at the moment. But also we've seen the changes in ratios. So maybe, something else is happening, but not much data is there. So more data is needed about this. Maybe, as I said, we might have new important mycotoxins. We have all these drought-tolerant fungal species that can grow pre and post harvest, and under increased CO2 conditions, they might produce things that are very nasty for our food, but we don't know yet, so more data is needed. The impact of climate change on crop physiology and how resistant they are to mycotoxins, there is little information around about that, only in few crops and for few species. Climate change, and especially elevated CO2 might stimulate the pest reproduction. So also, as I said, it will lead to higher damage. And when you have higher damage, you have higher potential of fungi contaminating your crop. But more data is needed. The predictive models that are used now to tell the farmers, when there is risk, when there is no risk, might not work properly, because as we have seen, when you add CO2, the prediction completely changes. And definitely the acclimatization of pathogens might play an important role here. But there is really not much data around, so more data is needed. And, another question is, how is this going to affect to biocontrol strategies and the way that we now have to control our mycotoxin in food? Very little data is available. So I think, when we look at predicting those food safety and security problems, and we look to both to developing and developed countries, this food security and safety could be profoundly compromised under these new climate change scenarios. And I think one of the big take-home messages from me is that when we looked, and we are 10 years down the road looking at how abiotic factors and related with the environment affect mycotoxigenic fungi and mycotoxins, and I still have, the question for myself, do we know enough? And I always come up with the same answer, and the answer is no. There is much more that can be done and needs to be done to really understand how this problem is going to develop. And with this, obviously, this is not a work that is a man in a van. This has been done by a large amount of people, not only at Cranfield, but around the world. And I've mentioned some of the names here in this slide. I would like also to again thank the BMS for giving me this opportunity. And among these, I would like to cite Professor Naresh Megan here, and the people in the Applied Mycology Group, Carol Burek, and our previous colleague Esther Garcia that has moved now. And you know, I would like, before giving it to the questions, to announce for those that might not know that we have the coming BMS Scientific Conference, with the motto Fungi and the Environment, here at Cranfield University on the 4th, from the 4th to the 7th of April. So if you want to know more about this topic or any other topic related with mycology, if it's research more lab-based, or if it is more field mycology, don't hesitate. Come this year, because we are celebrating the 125th anniversary, and the whole society is going to be here at Cranfield celebrating. And with that, I would like to thank you all for staying tuned, and I'll be very happy to take any questions that you might have.
Fantastic. Thank you so much for what a wonderful, wonderful talk. I can see some questions coming in now, but I was just going to say, just absolutely wonderful. And now that I've mumbled a bit to allow people to have questions, I should pass you over some questions. So we have one from Aditya Pankaj, apologies for the name wrong there, who asks, what is the effect of crop rotation as per the climate change can be helpful to reduce mycotoxins contamination? So what is the effect of crop rotation?
Yeah, so crop rotation, it depends on how the rotation is done, but it's very interesting that crop rotation is nowadays one of the ways on reducing the amount of toxin that you'll have in your crop. And this is because at the end of the day, this fungi survives in soil. And in soil, they have to, they can survive during the winters, thanks to living in crop residue. So what happens if you don't have the right rotation, or you have multiple times the same cereal, like for example, maize, the inoculum that you have for the following season is building up and building up and building up. And then you have much more problems. And this could be accentuated by climate change, because in some occasions when you have the right conditions, a little bit more drought, then the fungi kind of reacts to this. You know, they want to fight and defend themselves. And one of the ways that they have is produce many more spores and try to reproduce much faster, in such a way that they are sure that they will be able to survive in the next season. So it might be a problem. And definitely, I think you said Richard, but definitely Richard, doing the right crop rotation will help to reduce the mycotoxin. In fact, it's one of the good agricultural practices recommended to reduce the amount of toxins in your final in the cereals.
Thank you so much. We've got comment from Gavin that says thank you, but not a happy future scenario. I hope he's talking about your talk and not about the BMS conference coming up, because I think it's a very happy future scenario, but I'm not sure.
So
we've got
one thing that I will say to that comment is that, and this is what I always say, we need to trust science because of that, you know. So now we are at the moment in which we are pointing out you know, what are the potential problems, and the idea of this is this gives us like a kind of platform to start studying how to fight this and make that scenario that we put really never really get there. So put new control systems, understand the way in which we have to tweak the control systems that we use, the practices that we do in the field, in order to not having to arrive to this point in which we have that increase in toxin.
Oh, thank you. We have a question from Janet Quinn, who has been on the talk today. Says, fascinating talk. Thank you. So regarding the carbon dioxide induced increase in mycotoxin production, is there any evidence that carbon dioxide is acting as a signaling molecule, directly stimulating aflatoxin synthesis?
Thank you, Janet. That's a great question. This is one of the big questions at the moment because we've had different trials and different things we thought that we could do to figure out what is really the target of the CO2, what is really the CO2 doing. But as far as we know, we haven't been able to identify a single point. It appears to be activating multiple things in the fungus. It appears to be activating different pathways related with oxidative stress, and those appear then to be affecting the production of secondary metabolites. In fact, I haven't shown the data, but we have a comparison between the genes that are switched on and off between high CO2 and low CO2, and they are related sometimes with reproduction. They are related with secondary metabolite production, but we haven't been able to spot on. And the problem that we have is because of the way that Aspergillus flavus process CO2, we had this brilliant idea of putting some marked CO2 in contact with the fungus and then track those molecules that are produced with that CO2. But in the way that the CO2 is processed in Aspergillus flavus means that CO2 goes in and then the same CO2 goes out again. So we couldn't really track those molecules, although we tried, but with no luck. So very interesting question. We are on it.
Good to hear. We've got two questions that are quite similar, so I'll give them at the same time. So Jane Haydecker asks, is there any research on plant breeding to try and mitigate these effects? Similarly, I had a question from the BMS office, whoever that might be, who asks, is there an argument for genetically modified crops to grow quickly and require less water? Or is reducing aflatoxin argument for this? So is there any argument for plant breeding or genetic modification?
So we have some problems there, and I'm going to mention this. So the problem is that when we have the breeding systems for our crops, mycotoxins are not a thing that has been taken into account in the breeding programs. We've been more looking at, only the disease, for example, in wheat, the Fusarium head blight. So we are looking for reducing the Fusarium head blight, but not looked at how much toxin is produced by that fungus. And the same happened in maize. We're looking about increasing production, make it resistant to water, to drought. It might have some effects, but the breeding hasn't been done in that direction. And then what we might end up is with, for example, potentially kind of maize that might be more resistant to drought, but still be less resistant to the disease, or that might still have the toxin at the same level. What we have seen, for example, and that I can give you specific data, is we have compared in Brazil through our student Alessandra, now a doctor back in Brazil, that what we did is we compared the contamination of this fungi between GM and non-GM maize, and we looked at GM maize that had been introduced the BT gene to avoid insect damage, and also another gene that was making the outer layers of the grain slightly thicker. And what we saw is that, in fact, those modifications were reducing the amount of aflatoxin accumulated in the grain. However, that might be not exactly because of the genetic modifications that have been done. It's because, for example, if you have less pest damage, then there is less opportunity for the fungi to go in. So it's not that those genetically modifications are affecting directly the fungi. They are affecting the environment where the fungi is growing. And although they are not doing anything against the fungi, you reduce a little bit the amount of toxin because you are kind of hampering the entrance of the fungi in your grain. I don't know whether I gave the response. So that will be one way to go. But as I said, nothing has been done directly targeting the reduction of mycotoxins. It's more about yield and diseases that will affect yield. Nathan, you might be,
I am. Sorry about that. I said thank you. We've got some more questions, if that's okay. Oh, hello. Oh no. Angel, are you back? Angel?
Yeah, I'm back.
Brilliant. Wonderful. Thought we'd lost you. We've got a question from, and again, apologies on pronunciation here, Renos Muzuas, who says, thank you for a wonderful talk, but interesting and somewhat alarming talk. With increase in organic farming on crop rotation, is there any work on how mycotoxin fungi will react to fungicides in the future, especially if they can survive in soil?
We are trying to develop some projects in that area, mainly in the one in soil, Renos. So very interesting question. There is not much data, but as I said, is one of the main areas in the life cycle of the fungi that we think we could target some of the research, because at the end of the day, if we can reduce the contamination of the soil, we will be able to reduce the potential contamination of the crop. Then, I think the other question was if organic versus normal or crop rotation. So in terms of mycotoxins, in general, if you ask me, I'm not very fond on organic, because normally what will happen is that we will have the organic, and then, as I said, mycotoxins are still produced even when you are not seeing the production or the growth of the fungus, when you still haven't seen a moldy product. And most of the products that are used in organic farming are fungostatics, not fungicides, because the fungicides are normally more like chemicals. And then what we've seen also is that depending on the environmental conditions, and with climate change, this could get worse, is that when you try to hamper the growth of the fungi by using fungostatics under certain environmental conditions, the fungi again fights back and produces more toxin. And not specifically fights back with the toxin, but fights back producing a whole range of metabolites to protect him or her. I regard the fungi as him, to protect him against the environment and those bad conditions. They fight back with a set of things, and between, among that set of things, they launch those mycotoxins, and that might be against what we really want. So doesn't happen with all the toxins and in all crops, but it's something that we need to consider mainly if we want to move much more to organic. Another good thing is that we'll have data, for example, if you go to organic and you do biocontrol, we have data that shows that current strains that are used for biocontrol purposes, when you put them together with the other fungi that is producing the toxin, and you do the challenge again under climate change conditions, the one that is supposed to be reducing the toxin is not able to stop the toxin production anymore. So for Renos, that said, it doesn't look very good. It's even, could be worse if all the control systems that we have start not having such a big effect. I think with that, I will have answered the question, I guess.
I think that's a very thorough answer, and thank you so much. We've got a couple more questions left, if that's okay. Yeah. We've got another one from a teacher. I think the second part's already been answered regarding genetically modified plants, but also asks, temperature-based study and carbon dioxide concentration can be performed for other mycotoxins, such as iniatin. But if the level of carbon dioxide is increased, then temperature should be increased so that we can correlate the data if any temperature study is possible for any mycotoxin. So I think that's one about the co-studying of carbon dioxide with temperature. Is that showing any effects?
That's what we did. That's why when we looked at the scenario that will happen in 100 times, we try to put +2 or +5. In some areas, we put +7 because in the case of Aspergillus flavus, it drives when there is a very high temperature, and it will drive in countries like Italy, for example. They had a big outbreak of aflatoxin in milk that was supposed to be for cheese, and they had to throw away all the milk. And we always have the combination, because in fact, when you look at the climatic models, one of the reasons for the increase in temperature is the increase in CO2. So they always have to be hand by hand, and in all the research that we've done, they go together. With regard to eniatins, we don't have any data, but those are some of the emerging ones that people is looking at, maybe how important they might be, because they appear to be in many food commodities, and yeah, definitely will be interesting to look at them in the future.
Fantastic. So we've got last two questions. I'll start with the slightly more pessimistic one first, from Rua Zhao, who says, "Thank you, Dr. Medina, for a very interesting presentation. Could we witness in the future changes in mycotoxin regulations due to climate change, as the crisis that happened in Serbia that led to a change in AFM1 regulations to reduce food waste?"
Definitely, yes. The problem that we have is that the changes in regulation in the EU or now in the UK might be as slightly faster, but they are very slow, very slow. They require a lot of data, and things are changing very rapidly towards when we move in time. And definitely, I think that one of the main concerns will be, if we want to protect the consumers from eating this type of compounds, is the legislation going to be able to adapt, rapidly enough? Because, for example, what I was showing is that cyclopiaxonic acid might be even more important than aflatoxin if the amount that is produced increases a lot. So is the legislation going to be able to cope with those quick changes? And yeah, I know about the problems in Serbia with aflatoxin M1 in milk, and for sure, if this is still happening, and you can see, the risk of getting these very dry summers that we've had in the last decade keeps increasing, there will be more and more risk of having these sort of episodes around Europe. Because the problem is that in the case of aflatoxin, it was always considered a very important risk in subtropical areas. But now we can see that even in Europe, we are having a lot of hits with aflatoxins. In Spain, they are having a lot of problems with dry figs. They had problems with maize. We had the problems in Serbia that nobody was expecting. In Italy, places where aflatoxin, you could have thought it was impossible 20 years ago. And now we have to throw liters and millions of liters of milk in some occasions in some countries because of this. So I think, this is one of the main drivers that kept me pushing to do this type of research, although we didn't have a project about it, specifically a project of, so a lot of research that we did on the side, with students and for fun. But it keeps us going, seeing, all the things that were happening around us. And then we were really proud of everything we were pushing through and publishing, because it was kind of revealing, and it has opened a lot of different areas and a lot of fantastic discussions about science and mycology.
Fantastic, and what a fantastic positive spin as well. There's another question snuck in, but I'll give them both together, just because why not? So we've got one from Renaud Muzaras, who asks, are there robust analytical tools to detect mycotoxins in food crops?
Yes.
And then, yes, fantastic. And we've got one from Andrew Darby, who gets top points for bringing for most mycological question, who asks if you could make drought or carbon dioxide resistant modifications to original fungi, such as non-aflatoxin producing Aspergillus species, might it prevent a takeover by the aflatoxin producing fungi?
No, so the problem is, in fact, Andrew, that the fungi is very resistant to drought and very resistant to CO2. So these changes that we see when the problem is when the drought happens in the field, and then when you have a fungi that needs water to live, and another one that is very happy when water is disappearing. So what happens is that the one that needs water cannot grow anymore, and then is taken over by the other one that likes to live in drought conditions. So in this case, it won't have any effect. Aspergillus flavus is already very happy doing that. And it's really, really difficult to, for example, make more drought-resistant fusarium, because the machinery that the fungi utilizes to adapt to drought is very, very, very complex. And, it requires the production or the synthesis of many, many molecules that are called compatible solutes, and and then they have a controlling system in which depending on what is the water, they change from one solute to another solute in such a way that they will never get dry enough. So it will be a task of biblical proportions, really, to try to do those changes. It will require many years.
We've just had one question sneak in, which I think we'll call the last question, from Eric Janker, and it's a very one. It's a question of conservatism. I think you scared people somewhat, and he asks, is there any mileage in processing the food to remove mycotoxins?
Okay, Eric. So, as I said, the problem of mycotoxins is that they are very heat resistant. So although you cook, you boil, or you roast, for example, peanuts, the reductions might be only in the areas of 15% or 25% as a maximum, and very tiny amounts of it is very toxic for the people. It is true that, for example, with grains, when there is the case that the mycotoxin might be concentrated mainly in the outer layers of the grain, there is the possibility of, when you are milling or polishing the grains, I'm speaking, for example, about wheat, or that can be contaminated with DON, for example, or deoxynivalenol, or rice that might be contaminated with other toxins like citrinin or cratoxin A and aflatoxins. When you take those outer layers, you might be removing big proportion of the mycotoxin. So there are some potential there. However, if you have it, you have a problem.